Okay, so it's my great pleasure to welcome Dr. Lawrence Bramham from Rothamsted Research. And Lawrence actually did his PhD here at Warwick Centre with John Walsh. So it's great to see him here virtually, at least again. And Lawrence has taken on a new role as head of the Rothamsted Insect Survey. So it's a great opportunity for him to talk to us all about the insect survey and about what he sees for his new role. So over to you, Lawrence.
Thank you very much, Rosemary. So what I'll do today is talk for about 45 minutes, hopefully, and then we can crack on with some questions. But I was viewing this very much as an opportunity to give a bit of a holistic overview of where things are now in terms of the Rothamsted Insect Survey and a bit in the future. But also, this is a little bit of a tricky talk to do in that, in one sense, I'm talking into the void, but also I don't exactly know who I'm talking to. So there's a range of potentially pathologists, entomologists, ecologists, agronomists, and various other ists online. So I can't really engineer this towards any specific target. But in that, as I started to put these slides together, I realized that actually that is a major strength of the insect survey as a whole, in that it provides such a cross-cut across the entire agricultural sector. So whether it's, as you see here on the right, rather virus-infected wheat fields, exploring the actual vectors of things like viruses, the aphids, or the actual expertise and the hard graft that goes into the actual infrastructure of the insect survey behind the scenes. I'm hoping to cover all those aspects today, and hopefully there'll be a bit from this that you can take no matter where you're coming from, whether it is from a more agricultural or pathological or entomological angle. So what I have also got today, rather than just talking into the void, is a quick Mentimeter. I did this last week with some agronomists. It's a very simple ranking exercise as to where you see the priorities are for the insect survey. So if you do that in the background, then hopefully on the last slide, we should see the results of this and actually be able to compare it with what I saw with the economists. So it'll be interesting to see if you actually come out with the same priorities. So in terms of today, I'm going to cover three main kind of overarching topics. Context, very much around Rothamsted Research, where things are at the moment, because a lot of you might not necessarily be aware of Rothamsted Research, and those of you who are might be aware that we've undergone a major transformation very recently, so I can give you an update on that. Then leading on to outputs and research from the RIS side of things, or as I prefer to, the RIS, Rothamsted Insect Survey, from the data side of things, this Insectageddon, the Armageddon of kind of insects and insect decline that we're seeing, as well as predicting the future, and then leading on to challenges and opportunities. And this aspect is a little bit more RIS adjacent in that it's some other work that I've been pursuing, all of which has been supported by the Insect Survey. So Rothamsted Research, this is our main campus over at Harpenden. You might be aware of our kind of usual taglines of global science, lasting benefits, building on over 180 years of science excellence. We're based, as I say, in a couple of different campuses. So the main one in Harpenden there, just north of London, as well as Warwick, our other kind of sister secondary site, as well as some field stations at Woburn and Brinsbarn. But the real point just to highlight here is that we've got that tagline of 180 years of science and excellence. What really are we now, and what are we for the future, especially after this major transformation that Rothamsted's gone through? Now, I've done an absolute cardinal sin here when it comes to presenting slides. There being a lot of text on here. This is mostly just for your reference later. But what I really just want to highlight here is that with our major restructuring, we now have 6 strategic science teams, covering various facets of UK agriculture. But the idea being here, that they're more for managerial purposes, and there's actually supposed to be a lot more interconnectivity between these. 2 of which to really focus on are in the bottom left, the Agro-Ecosystem Health and Pest Management team. That's run now by John Storkey. That's where the Insect Survey sits and who I'm officially line managed by. But then there's also the Translating Biotic Interactions, which is run by my former line manager, Kim Hammond-Kosak. And between these 2 areas, this is where there's a lot of synergy and involvement of the RIS. So I mentioned the RIS. It is one of these national bioscience research infrastructures, which are all aspects invaluable to the innovation community. We're very proud at Rothamsted to host 3 of them: the long-term field experiments, Northwick platform down in Devon, and also the RIS. And this is really where to first introduce what actually the RIS is. So if you to say in a nutshell, it is just this. It is 2 trap networks across the UK that have been running since 1964, so over 60 years. During this time, they've mostly focused on monitoring aphids, as well as large moths and many other migrating insects. And you say it quick, but I don't think this can be understated, or overstated rather. It is the most comprehensive and, more importantly, standardized long-term data set on insects in the world, with a wide range of scientific and applied uses. And what we're incredibly keen to do is actually, alongside all of the other embryos, is to build capability, collaboration, and culture. So aside from actually just giving you a bit of information about the RIS today, this is a little bit of a call to arms. If there is anything of interest that I flag up, do reach out. We're so keen to actually connect with you, do some research together, and take this amazing infrastructure and resource forwards into what is now modern technology developments, etc., etc. So actually going now into the context and the ongoing value of the RIS, I really love this phrase. I think it was coined by Richard Harrington, one of the former leads of the RIS, foresight from hindsight. And there's a brilliant paper there in the bottom left that covers the RIS in more detail. But this aspect of foresight from hindsight, I think, is a really cool aspect of the RIS, having been run for so long, and not just from the angle of being run for so long, but it's stayed relevant even across the 60-year plus lifespan. It's worth looking back to actually where the origins of the RIS started. So it goes back to what are called the Cardington experiments. And these, back in the day, in the 1950s, were run really by the pioneers of a lot of insect and aphid migratory research, where prior to a lot of health and safety red tape now, they strapped a whole series of suction traps on these dirigibles, sent them up at over 1,000 feet, and the entire aim was to understand the dynamics and mechanisms of aphid migration. Following this, in the 1960s at Rothamsted, there was a series of these scaffolding experiments where, again, suction traps were put at various heights, and the idea here was to identify optimal sampling for aphids and other key migratory insects. Now, it was at this time that probably one of the real precursors and kind of kicks up the motivational route for actually getting the RIS started was this amazing book published by Rachel Carson. So this explored the prophylactic use of insecticides, especially DDT, and is recommended reading for anyone who is interested in this area. To go into a bit more of a contextual timeline of the RIS, this is insecticide use, or rather the development of resistances over time, from some excellent work done by Chris Bass over at Exeter. So you've got starting off with organophosphates back in the 1940s through to neonicotinoids, which are more commonly known of today. And to give you a context of timeline, it was back in the 1950s, as I say, where those Cardington experiments started. That book, The Silent Spring, was published in 1962. The RIS started very promptly after, really to explore the prophylactic use of insecticides and their impacts upon aphid migration. And then, since then, there's been a whole series of technologies developed, such as things like PCR. And really, if I'm going to highlight anything of note in this slide, it's really just to highlight that since that original start and the pioneering of this sort of resource, there have been so many technological developments since, and all of these samples have been saved for a future use. I very much see we're now at that future, and it's a really exciting time to actually be leading this survey and hopefully collaborating with some of you who are online today to see where we can take research forwards. One route is through molecular testing. So Rosemary is very familiar with this as the author of this paper. This is looking at Mysis persicus, one of the key aphids and almost a poster aphid, as it were, for insecticide resistance. And you can see over time that populations have changed, as they've been tested against things like modified acetylcholineesterases tests, as well as pyrethroids and superpyrethroid resistance. And it's the necessity to keep monitoring the aphids that's really kept things going. And so this stage, I really want to introduce the infrastructure of the RIS. So it is a network of networks in many senses. On the right, we've got a video running which covers our light trap network that's been running since the start. You'll see over time occasional dots pop up. This will just show where and when we've actually had light traps. These have been monitoring every night that they've been in operation for any light attracted insects. So mostly Lepidoptera, things like moths, and ultimately contributing to greater knowledge of both biodiversity as well as loss of moth pressures even, and other agricultural aspects. It's run by a fantastic network of over 160 volunteers at what is now 58 locations, and the data here is invaluable from an ecological angle. On the aphid side of things, we have the amazing suction trap network. So we have 4 sites that are run by our sister organisation SASA up in Scotland, at Inverness, Dundee, Gogarbank in Edinburgh and Ayr. Meanwhile, in England, we have at Newcastle, York, Curtin, Broomsbarn, Little, East Malling, Rothamsted, Silwood Park, Wellsbourne, where Rosemary is, of course, Harper Adams, our newest trap, Hertford and Starcross down in the south. And at each of these locations, we've got these 12-metre tall suction traps that day and night are constantly hoovering in aphids and other migratory insects, captured and preserved, and then retained for future analyses. So these are emptied daily by a team of, again, volunteers. They're ID'd back at base at Rothamsted. We have an amazing team of entomologists who are going through each and every capture and identifying them down to a species level, specifically on the aphid side of things. But these are available for a whole series of analyses, whether it's modelling of the captures and also taking those samples and looking at them from a whole organism biology aspect or molecular testing, you name it, we've got it as a resource ready for you to use. And then that's just to highlight where we're at the moment in terms of the light trap network. Now, this is really where I introduce the pillars of the RIS. So this is kind of really where our research aligns. So whether it's in climate change, spatio-temporal ecology of the insects, trophic ecology, and monitoring and population ecology. And here you can see it broken down into various aspects. Today, I'm really going to focus on these. So trends, pest outbreaks, a bit of forecasting, and vectoring of viruses and insecticide resistance. But if I just go back for a second, this does link as well with our overarching objectives of detecting and communicating the prevailing UK insect populations, as well as highlighting any emerging threats, qualifying population changes from a genetic, phenotypic aspect, using a combination of molecular tools, as well as entomological expertise. One thing we're really keen on doing, alongside actually just capturing samples and then getting them available for use, is actually providing the insect ID training. So if you do have any desire to learn more about the insects, whether you're from an agronomist background or research background, do get in touch, because we're happy to host anyone and everyone and provide you the skill sets to actually have some upskilling in this area. We have a sample archive, as I say, for whole organism study and molecular exploration. But in addition to this, we also have a digital archive, which we'll go on to discuss shortly, that's got a range of structured query language systems and various infographics for exploring the data that we generate. This is the amazing team. So I'm the head of this team, but really is down to the expertise, whether it's Louis, Mike, Alex, Tracy, Rob, or Harry, all of whom are phenomenal entomologists, or in the case of Mike, an entomological engineer who keeps our RIS suction trap network going. And it's really down to these folks who ultimately get a sludge of what is a mess of aphids and other insects. They separate them out, ID them, and equip us with the knowledge of what's actually out there, when and where. I can say it very quickly, but it shouldn't again be unstated. Our sample archive now contains over 100 million specimens, all of which have got linked metadata, whether it's location, the weather at that time, and other aspects. And per year, just on the aphid side of things, we collect close to 1 million. So, for example, last year, we collected just close to 920,000 aphids, each and every one of them ID down to a species level and archived, available for study. But it's not just aphids. So this is some work from Druid, an affiliate of RIS. You can see here, that's kind of the breakdown of the sort of thing that are collected. Alongside collecting aphids, we also subset this into what we call bycatch. So if there is anything here of interest to you, do reach out again. It's a phenomenal resource, and we'd really like to get you on board with doing project collaborations. And This is a snapshot of our archive, again, just giving a taste of, or a flavour rather, of what we've got available for study. So to go into kind of what is available and a few stats, as I've previously mentioned, we've got over 50 years of sampling from about 1974, when the more modern style of sample preservation was set in. It comes out as about 240 samplings per year, with over 160,000 samples available to study, where a sample is effectively a capture. And for each of those captures, we average about 800 invertebrates per catch. And as I mentioned before, we've got over 100 million specimens ready to go, all of which stored in 95% ethanol. Varies a bit, but well over 95% of ethanol in that fraction, plus 5% glycerol to keep those samples preserved long term for whatever study is needed. We also host an excellent external website, insectsurvey.com. That's what it says on the tin. It's an access point for looking at data, but also getting in touch with us as to whether you've got any projects that you'd like to pursue. So if you do go on the website, this is what you'll be met with. It's the insect survey breakdown, whether it's aphid data or moth data. To drill into this a bit further, you can select, let's say, for example, aphid data, select your area of interest, and then get what is basically near real-time data on what the aphids are at the moment, as well as also historical. So for example, here, looking in the North England area, you get these kind of pie chart icons. These are populated with information. So depending on what's going on at that time, so for example, here there's a blue highlight. This means that it's decreased from last week, but then also the pie chart that fills up gives an indication of how it compares with either the 10-year mean or previous values. So it's a great resource if you want just a snapshot of what's going on at the moment as far as aphid pressure. And this, of course, has wider impacts, whether it's for just research, but also agronomists wanting to know what is actually out, where and when, so they can spray against it and ultimately control things like viral disease spread. There's also weekly bulletins with linked insights. So every week we send out this sort of information, and this is updated on the website alongside an excellent overview provided by Alex Greenslade of what are the real notable changes. This was a particularly busy week for the RIS back in June earlier this year. You can see in some cases, for example, Midas persky, which I mentioned before, is the poster aphid. In some areas, we're getting over 1,000 aphids in one single week in one single trap. So this really does just give you the scale of what we're actually collecting and the potential for this to be taken forward and used in new and exciting ways. There's also a lot of data visualizations on the website. So for example, here again, the Midas persky, although this time purely at Wellesbourne, where rosemary is. This is what's been collected over the last year. So you can see the usual kind of peaks and troughs over the year. So a temporal resolution of when actually to be concerned about aphid pressures, as well as actually also how this year is comparing to others. But then we also have this as raw data format. Just reach out to us and we're happy to provide data as we can. One thing we also offer, which is excellent, is a free weekly texting service. So I'd recommend signing up for this. What this effectively does is each and every week you get a text on a Saturday which shows you the number of insects caught in the closest suction trap to you, based on like specific ones of interest to you, as well as the number of insects caught in the local area. So for example, here in East England. And this is really an ideal route for actually equipping farmers with knowledge of what's out there, when and where to be concerned, and when to actually start spraying or take other IPM-based approaches to resolving the issues. So that's kind of the RIS website, but just as kind of a little bit of a break and to highlight some of the notable facts and figures. So from our suction trap network, we've actually collected 478 distinct aphid species. So it's not just a handful of aphids causing problems. There's a huge number out there, and it's really a testament to, as I say, the expertise of the team here, that all of these, no matter which one they get, down the suction trap, they can ID. So that's providing great insight. In terms of the most aphids that were caught in a single day, it was an astonishing 51,136, although the vast majority of these were overrepresented by a single aphid species, the rose grain aphid, back in 1979. And then taking a switch to our light trap network, the number of distinct moth species that we've identified is over 1,500, and the most moths that were caught in a single night is 103. So again, it just gives you a sense of the scale of actually how many insects we're capturing, the diversity, but also to highlight this as a resource. Going back all that time to when actually the pioneers set this up, they had no idea about the new technologies we'd have today, things like machine learning, AI-based detection. We're now in a place where we can actually take this phenomenal resource forwards and explore it, as I say, in new and exciting ways. So that leads me on to really discussing a bit about the outputs and research now that I've given context of the RIS. And I think the first thing, which always is flagged up whenever I have chats with agronomists or folks at cereals or the general public, is the insect again, the decline that we've seen in insects, both anecdotally and in various other research studies for what can be considered well over 100 years now, and as I say, anecdotally for about 150. This can be caused by a whole myriad of factors, things like habitat loss, deforestation, and the conversion of landscapes for agronomy and more human uses, ultimately reducing the habitats available, the use of pesticides, incorrectly or correctly, but that widespread use potentially having impacts and contaminating the environments beyond the target of use. The big C, so climate change, ultimately changing the way that these insects are behaving, both on a regional, but also much more geographical level, and even aspects like light pollution, so the use of more artificial lights, actually disorientating and potentially harming the movement of insects. Now, when I do speak with various folks, of course, the anecdotes come out, things like there's fewer bugs on my car windshield, which I completely agree with. There used to be clouds of insects overhead. I can't remember the last time I saw a bumblebee, and my personal favourite, which pops up more than often, is that it's your damn survey hoovering up all the bugs. It's your fault, which always makes me chuckle. It could actually be considered a little bit true, although not. But when you look at the actual number of aphids and other captures that we're capturing, although this is a drop in the ocean when it's concerned to actually how many insects are out there. But some amazing work done by James Bell, the former RIS lead, looked at historic data from the suction trap and light trap network and analysed over 24 million aphid moth captures to see whether things had changed over time. This was based on 25 suction traps. So there's actually 24, but there's one which is a bit of an overlap in Edinburgh area. So 25 technically different traps. And this covers what can be seen as 725 site years and 112 light traps. Again, about 2,500 site years, all of which spanning over about 47 years of data. So this really is, as I kind of alluded to over and over again, an amazing resource for looking at ecological change over time. But what are the outputs? Now, it's really quick and easy to say, oh, we've got X percentage drop, but these sorts of explorations really need a lot of nuance. This slide nicely summarises that sort of challenge we're faced when actually thinking about insect decline. So, for example, we can have things like a shifting baseline effect. When do you take as the start of when there was a normal presence of insects? You can have years which have kind of extremely high versus extremely low. So that snapshot effect aspect, do you see a sudden change which isn't actually representative of the drop? High interannual variability and left and right censoring, so omitting certain years. All of these have to be factored in. So I would say that any and all data on this topic does have to be taken with a bit of nuance. And do please go and read the papers to get that rather than just a quick snapshot. But that said, in terms of a quick long-term linear trend, aphids come out as about minus 7.5%, although this is, based on the stats, not significant. Meanwhile, moths and other Lepidoptera, it is much more notable and significant, with a minus 31% drop in prevalence detected. This flow is purely looking at linear trends, and in order to actually get all of the nuances of the data a bit more, there needs to be more convoluted analyses done For example, these such as flexible, non-linear, generalized additive modeling covered by James in this paper. So you can see here, hopefully, all of these almost like box and whisker plots. Between years, there's massive variability on the aphid side of things, which does pose quite a lot of challenges in terms of actually modeling aphid movements and behavior and predicting the future threats that we face. But yeah, you can see there's quite a little bit of variation. Meanwhile, on the moth side of things, there has been when you do it in this format, a significant 48% reduction. But notably within this trend, there are periods of significantly decreases followed by stabilization events where moths and other Lepidoptera can recover. But overall, short answer, we are seeing declines in insect overall, and the resources such as there is demonstrate that. But as I said, there's always caveats with this data, and for example, with the aphid side of things, over half of all the aphids represented can be covered by just 3 species of importance, including R. paddy and Stolia novini, which I'll discuss a bit later. There is the aspect of, well, what's really driving this sort of change? Is it things like climate change? Is it habitat? So really, if we do look at habitat first of all, these are actually 2 of our light traps, one in France versus one in, well, I'm not sure exactly where this is, but you can see the soil surrounding. And I know where I'd prefer to be, but at the same time, I'm not a moth. So if we do break this down into things like coastal, parkland, mixed, urban, farmland, scrubland, moorland, and woodland variations, these are the sort of trends you see. There are 3 key points of highlight. So for coastal, urban, and woodland, the trends can definitely be described as significantly declining. There's some areas like moorland where there is a statistically significant difference, but it gets a little bit tricky and nuanced. But quite interestingly, farmland, it's relatively stable, but with these massive fluctuations year on year. So there's a lot of exploration that needs to be done here still. And this data was based on up to 2016. So now that we are 10 years on, there's massive potential here to actually explore this further with the new technologies that become available. So do reach out if there's something of interest that you'd like to explore there. There are, of course, as well, winners and losers in all this. We can't just look at moths and other aphids in a single angle. So of the 427 species with abundance trends, majority have declined significantly, whilst there's some that have increased significantly. And then in terms of distribution, it's a mixed bag. Some have declined, others increased. There's just a lot to this. But a lot of this can be driven by climate change. So for example, if we look at things like the dingy footman, which has shown quite a nice consistent increase, and we've updated these analyses with the help of the UKCH through to 2025, you can see there's an increase in abundance, but also distribution on the right. And this is an important point to highlight, that what we're seeing increasingly is with climate change, insects moving more north in terms of their distribution. This is having a negative impact upon most species that require kind of more northern ecological factors like damp, cold conditions, but those that are enjoying the warmth, we're seeing movement up. So this is partially why it has to be done on a species by species basis. Taking a switch now to the aphids, we want to really be in a position where we can actually predict the future. So one thing that's a major RIS output every year is the forecasting side of things. So the best predictor of this in aphids is the mean temperature recorded in January and February. So early next year, we're looking at this and then modelling, and then across all the various sites of the RIS. For example, here, looking at this bird cherry oats aphid, Rothamsted paddy, we can predict when we expect first capture of aphids to emerge in suction traps with relative confidence. This is based, I'll skip that for now for the sake of time, but this is based on things like this. So historic work where a nice regression can be plotted against the mean temperature in winter and significant trends identified. The point here to flag though is that from an agronomic perspective, even something just as like a single degree plus is going to have implications for when aphids are going to be emerging and actually spreading things like viruses into the field. So for example here, a single degree plus has shifted the emergence of the aphids by a few weeks. This will have impacts. Combined with this, we can also model what's going on in terms of actually what aphid varieties and types and morph types are out there. So not all aphids will spread viruses in the same ways. Things like the serial colonizers versus non-serial colonizers are important. So here, for example, with one of the major serial colonizing aphids, Arpadi, we can see that there's this trend, again, based on temperature between December and February, as to how what proportion of those will be colonizing cereals later in the year. So this is another area of exciting research. This kind of leads me on to actually where I fit into things a bit more. So I'm, in a sense, not actually an entomologist, which comes as a bit of a surprise when you consider I lead the insect survey. But I come from a background, as Rosemary mentioned, of more on the virus and the genetic side of things. So I've done a lot of work in this area of UK crops and the pathosystems such as viruses spread by insect vectors, whether they be aphids or other things, and improving diagnostics. As I mentioned before, there's a huge plethora of UK aphid species out there, but for now, just focusing on 3 of importance. So the Arpadi, that's the cherry oat aphid, the English grain aphid, and Mysis persicae. And this is also a time to flag up what I think are a major, or how to phrase this, kind of feather in our cap, as it were, for actually getting data out there, which is the 5 GINs, the Genetic Improvement Networks. So these valuable networks are funded via DEFRA, whether they're VEGGIN, Origin, WIGIN, PCGIN, or SoftFruitGIN, covering the vast array of crops across the UK. And it was actually through the start of my PhD work exploring turnip mosaic virus and turnip yellows virus to aphid vector viruses in VEGGIN and Origin that I really got passionate about this subject. And now I am the deputy lead of WIGIN, where I work, or have worked rather a lot, on barley yellow dwarf virus, an important, again, aphid-vectored virus, mostly affecting wheat. And it's through this network that we actually interact with a lot of industry and identify the best routes for deploying new genetic resistances, as well as knowledge of what's actually out there. But all of this is, you're thinking, how does this link with the RIS? We ultimately need to know what is out there, where and when. And by actually forming networks of networks, linking with things like the DEFRA-funded GINs, we are improving the ability to know what are the major threats and how to actually tackle them head on. So that leads me on to challenges and opportunities. On the virus side of things, there's a whole plethora of UK virus vectors, whether they are aphids, the big one, but through to thrips, beetles, mites, leafhoppers and plant hoppers, which spread viruses in different ways. I've got a nice animation here on the right that shows the persistent route of virus transmission, where effectively an aphid in this example picks up a virus, it goes into the hemolymph of the insect, migrates to the salivary glands, and then from that point onwards, that insect can spread that virus for its life. And that's a major issue in terms of control. Skip that for the sake of time. In terms of control, we only have a limited arsenal available. One of those is insecticides, but I like a chart where you can interpret it in less than a second. This is very blue, and this just highlights the insect targets where stated for foliar insecticide application to wheat in the UK. And you can see at the moment, it is 92 to 95% aphids, and more aphids. This is causing major challenges in terms of the evolution of insecticide resistances due to high selection pressure. And this is bounded based on the actual number of insecticides we have available or what range of insecticides we have. So again, a very blue pie chart here. This is just showing the type of insecticides of which we are predominantly spraying or using things like pyrethroids, pyrethroid-based active ingredients, at least on the wheat side of things. In those aphids I mentioned previously, the 2 heavy hitters of spreading viruses like important ones like barley yellow dwarf virus, there's already resistance emerging. It's widespread in things like Cytopium aveni, and there's evidence now emerging of pyrethroid tolerance over in Ireland. And we need to get a bit of a handle on this to actually help manage what's going on. We can't also view aphids as just being the same. Different aphids will spread viruses at different rates. In terms of different strains, we need to know what's actually out there. Similar to a COVID analogy, unless we know the different strains and who's got resistance and the actual diagnostics working, we're in a situation where we're testing, and how do you trust what might be a false negative? So an angle which the risk can really jump on is actually improving the diagnostics, and that's something which I'm very passionate about. We do have angles like the deployment of genetic resistances, so genetics is a major importance, but if we don't know what's out there, whether it's the aphids, the inoculums, such as viruses, resistances, whether it's resistance to the aphids or the virus, how do we actually tackle resolving these challenges, as well as the resistance spectra? And a major point that gets flagged up time and time again, which is resistance or tolerance. So on the right here, I've got a bit of a flow chart. You can break it down quite simply in terms of the virus side of things, but this applies as well to other angles. So whether it's insecticides or aphid resistance, you can have a host or a non-host. You can have the behavior, so something which is susceptible or resistant. But where things really get convoluted is whether it's tolerant or sensitive. So you can have, for example, here, a situation where the virus might be replicating in a susceptible plant, but it's not giving a yield penalty. But then it raises a challenge of, well, can these tolerant varieties harbour virus and or influence virus strain prevalence? And this is an area that we really need to get a handle on and explore. If we look at vector life cycles across the year, there's an increase and a decline, then a build again. And historically, we've relied almost on winter as an annual population reset to stop the proliferation of aphids and the spread from season to season. But now that climate change is upon us, we're reaching this stage where there might be more warmer autumns and winters, more overwintering survival, and that can carry additional challenges, whether it's genotypes the aphids carrying over or actually the spread of viruses. No matter how we square it, whether it's elevated carbon dioxide in the atmosphere, elevated temperature, or drought, these are all going to impact aphid movement, whether it is the flight activity, the actual generation times, how quickly aphids are reproducing. And again, this puts the RIS in an ideal position to actually be central to monitoring these challenges and linking with things like the Met Office and other organisations for better modelling of the threats. We are seeing already things like this with the Met Office. So, for example, here we've got a mean temperature anomaly. So this is actually the difference that we saw in 2023 versus a 30-year period roughly prior. In autumn, there's a lot of pink here, including some red there, where actually there's regions that are higher temperature than they used to be. And more importantly, for aphid overwintering and the spread of viruses or genotypes from one season to the next, we are seeing more potentially overwintering in certain regions. And this is something we need to, as I say, really monitor for. In the interest of time, I'm going to skip this and just get on straight to BYDV testing. So on the BYDV front, we didn't really know what was out there. So this is a major virus of, as I say, wheat and other cereals. A lot of the diagnostics were not strictly designed for UK variants, and there's a lot of potential for misdiagnosis. So employing the RIS, what we did was we established a baseline for strain variation. If you look here, this is a breakdown of different strains across the globe, specifically looking at two of interest, so in blue, MAV and PAV. But you can see here, it's not just a single blob of red, a single blob of blue. There is variation across the globe, and we didn't know what was actually present in the UK. And this is problematic because a lot of diagnostics might be based on specific references, for example, this one here and this one here. So we took RIS samples since 2020 and have been monitoring them for BYDV presence in collaboration with AHDB. These are the results that we get. So things like percentage of BYDV. So BYDV is consistently detected UK-wide, but between years, there is a switch of actually the incidence. So even in actually roughly the same month, between one year versus the next, we can see massive variation. This is something we need to model better but we have the resources, as I say, available to do this. The virus strains are also not strictly conserved geographically, so this is just a snapshot of what was actually out there. And you can see there's certain regions like Hereford and Starcross where if you squint, they're roughly the same, but other areas like York where we're seeing massive differences over time. And this is, of course, going to have implications for resistance screening or germplasm development. So actually virus testing is another area that we can really take those forward. Skip down in interest of time, but where we actually can really take things forward and where I'm wanting to highlight there is as a major resource is actually the sequencing and the molecular diagnostics side of things. So just as a subset of 2 years of data, we explored actually the BYTV variation across the UK and could identify what are called these SNPs, so UK strain-associated polymorphisms, but more importantly than this, actually identify regions of conserved DNA or RNA rather in this case of these viruses. This has allowed us with the support of the British Society of Plant Pathology to develop assays. So for example here, we developed an assay which, long story short, you can take an aphid, pop it in a tube, give it a squidge, and by heating it up, see a colorimetric change that will give you a qualitative indication of whether you actually have BYTV in your field or not, which I'm hoping is going to be a bit of a game changer for those who are suffering with this impactful virus. But that's just one example, a single virus amongst a huge number of aphids that we can explore and do amazing things with. So I'm going to actually skip this section in the interest of time because things are ticking on, but we really are looking forward as to actually what is on the horizon. So on the instance of wheat, we know that there are 55 impactful viruses. There's 16 which have a known incidence, whether it's current or historic, in the UK, with a reasonable proximity, i.e. in Europe, and a vector component. 3 of which have the ability to really move and will be changed by climate change, and so we're focused on those. And these are kind of where those things are at. So all of them currently are in Europe, all of them currently have the vector in Europe, and we're now exploring what is actually present in the UK. And this is an area which the RIS is well equipped to explore. We're already exploring barleyvirus G on the aphid front, trying to confirm records whether they are present or not. Wheat dwarf virus, a major emerging virus for wheat and other cereals spread by this leafhopper, is now confirmed, and we've got diagnostics up and running for that. And then really just to finish off today, I want to have a bit of a future vision of where I really see the risk going, which is through a lot more collaborations, for example, with Met Office and other entomological radar. This, for example, is a vertical looking radar we've got based at Rothamsted near our suction traps that is constantly scanning overhead. There's lots of potential for remote detection, as well as artificial intelligence-based detection, such as these cool pieces of kit that we're going to get on site shortly that can automatically identify aphids and, well, potentially aphids and Lepidoptera, with the idea of generating more and more data to reinforce what resources we have available. There's a need for more molecular assays, but we do have a lot of those assays available. For example, here on the insecticide resistance front, this is a recent paper published by Stephen Foster and myself and some others, looking at what we're seeing across the UK, especially in context of insecticide resistance failure. But what we're really wanting to do now is ramp this up and be testing in a lot more high throughput fashion. And this is example data here for the neonicotinoid side of things, where we have now confirmed the presence of neonicotinoid resistance coming through, at least in a heterozygous form, in certain aphids. This, for example, is in that Mysis persicii, the peach potato aphid. So it really was a whistle-stop tour, trying to keep this to 45 minutes. We do have a lot of challenges, whether it is things like shifting virus strains, which are the major threats associated with aphids in the UK, aphid populations changing over time, the big C of climate change and how we're going to tackle the challenges that that brings heads on, as well as actually robust resistance deployment, whether it is genetic resistance to viruses or actually aphids themselves, and ensuring that our current tools actually last. So we have all of these insecticides, as well as new insecticides coming on the market, as well as genetic resistances and other IPM methods. But all of this requires ongoing characterization to ensure that they will work long term and are actually reliable for what we've got currently in the field. But really, this is where the opportunities exist, especially from the angle of the insect survey. So the RIS is formed of such phenomenal expertise, excellent infrastructure and availability of data, which we're really keen for you to reach out and us provide access to, as well as the samples for wider research. The resources also exist for modelling changes to important UK insects and vector crop pathogen path systems, and also new tools, which we'll not have a chance to properly go through, but things like that high throughput assays and molecular diagnostics. So that really leaves me just to say thank you so much for your attention today. I realise it has been a bit of a whistle-stop tour, and I've been trying to cover a lot of different angles based on the plethora of expertise and also interests that are probably present in the audience today. But all Rothamsted Insect Survey staff and volunteers, both past and present, because it's been a huge institutional resource for such a long period of time. Rothamsted Research colleagues, of course, as well as industrial and academic collaborators, and continued assistance from the wider community, and of course, the funding which makes this viable, especially from UKRI and DEFRA. And I've got some contact details there. So yeah, thank you very much. I really appreciate the opportunity to speak to you today. And I think hopefully this should work. So this is the questionnaire outputs from when I spoke with the agronomists. So you can see here, the ranking is very much on the angle of diagnostics through to support tools. So unsurprisingly, more on the angle of what is actually impacting them in the field. And fingers crossed this works. Here goes nothing. Yes, so here we go. This is based on the respondents today. So number one is better modelling of the pests. So much more on the modelling side of things, more easily available data. So that's really good to know. That decision support tools, which is vital. Diagnostics coming a bit lower down versus the agronomists. But similarly, more trap sites and connectivity with industry coming down at the bottom. So that's extremely valuable, and I really appreciate it. But other than that, I'll stop sharing, and I'd love to answer any questions you may have. So thank you very much.
Brilliant, Lawrence. Thank you very much. That was amazing amount of content. Brilliant.
Yes,
so there
covered a lot.
you did, and you skipped some slides. So there are 3 questions in the chat if you want to look at those, or I can read them out, whichever.
Completely. I'll go chronologically. So Janet, can the light traps be used to monitor light brown apple moth populations so the occurrence of 2 or more generations per year can be identified? I think probably yes. I'll have to have a check of that specific moth, but any and all moths would tend to pick up in the light trap network, so we can explore that later. I'll make a note to have a chat with our main light trap organisers and see what actually we're collecting in terms of that and go forward. So please do, Janet, reach out to me and we'll follow up on that separately. Yes. Then Susie, so do the results of your insect survey get uploaded to a biodiversity database like NBN if you find any species that haven't previously been recorded in the UK? So we have had instances of new insect emergencies coming out, and as and when those occur, we put them out via publications. So, for example, there was a cryptic species of aphids that came into the UK a couple of years ago, and Alex Greenslade generated an excellent publication on that. And then what we're also exploring at the moment is the ability to have a standardized upload of our RIS data so that it's more accessible than it is currently. One issue with this is that we have a lot of quality assurance that goes on. Because we're getting aphids coming in so thick and fast, we have a lot of the time aphids that have to be checked later on in the year, pretty much in this kind of winter season. So we always have a bit of a lag as to what is actually live on the website. But I'm trying to get things into a place where we have that more structured so that we can go and see the 2025 data set, as it were, then and be able to exploring that in its raw format with inputs from ourselves on the support side of things. Janet again, hi there. So can you identify when potato and strawberry aphid populations are likely to be first seen in strawberry crops? I have to admit, I don't know as much on the strawberry side of things as I should, but I am in close contact with the soft fruit gin, as I mentioned. So I'll have a good chat with them when we actually have our catch up on the 16th of December in just over a week's time. Depending on which aphid species are the major issues, we can explore that. But my understanding is that a lot of the strawberries are grown under glass or in polythene. But please do correct me on that if I'm wrong. But on the potato side of things, that is an area of significant research, and we have a lot of tools and ability available to be monitoring, especially the miser side of things, miser and its movement and prediction across the UK. And then Gary, Lawrence, can you tell us about the manpower required to identify the trap catchers? Fantastic. And yes, also the aspect of the automation possibilities. So this is a huge area of potential development in the future, because as you've seen, we have a lot of aphids in the UK and a huge number that are coming down our traps. And a major bottleneck is taking those samples forward and actually identifying them down to the species level. Of our staff here, comfortably it takes over 5 years to train them up to the level where they can take any trap catch and be able to identify all of the species in there. There's some really cool technologies emerging, like automatic sorting of aphids alongside automatic imaging. And I'm in contact with some folks over in mainland Europe who are exploring that. I'm hoping to get some grants of a way to hopefully get that kit into our suction traps. On the light trap side of things, we're already exploring those. I think there was a photo on one of my slides, the kind of automatic monitoring of insect traps. We're going to get a few of those on site in early January. So there really is potential there to actually have automation. But the real problem is, although it can be seen as a problem, can be seen as an opportunity, we can't compromise the RIS as it has been. So as it's been a resource that's been running for over 60 years, we can't suddenly shift things overnight. It is going to be a period of time where we start to bring in all of these emergent cool technologies, work them alongside the RIS, and then see where there is the ability to do more remote modelling to complement the infrastructure that we've already got. But it is an area that's already underway, alongside also the modelling. So rather than just having site-specific models, so you know what's going on at Broom's Barn or Warwick, or we're wanting to get to a stage where we've got a much more holistic view of the UK, and almost being the case that you could imagine you pop a suction trap at a random GPS location, what would you get in that area? And so we're pursuing some routes for doing that at the moment. So I think that's all of the questions in the chat, but I'm going to look to answer any questions anyone might have in person, or if you do want to, just reach out to me on email. Yeah.
Yep. If anybody wants to put their hand up and ask a question in person, that's absolutely fine. I don't know why I put my hand up. What was I going to say, Lawrence? Because obviously the aphid data comes back in sort of almost real time.
Absolutely.
There's a week or so, but the moth data I think takes longer, doesn't it? Does it?
It does. I mean, the nature of that is because we have this, as I say, a phenomenal network of volunteers who are taking the catches every night. They're then actually identifying them in their own personal time and then feeding back that information, which we then upload onto our databases as and when we can. But there is a bit more of a lag there, partially because we don't know exactly when that data is going to be coming in. Meanwhile, for the actual aphid side of things, we have the kind of nice throughput system ready to go. And yeah, each and every week there is a team there processing all samples and uploading to the website. So we are running on almost a weekly basis. Every Friday, getting a bulletin ready to go. And for those of you who sign up for it, you'll receive that via email or text. I can see Gerard has got a question.
Hi, Lawrence. It's
Hello, Gerard.
Croft from the British Potato Trade Association.
Oh, fantastic.
You're probably well aware of the issues that we've got with potato leaf roll virus in the potato sector at the moment. Couple of questions. One, I understand from the Scottish traps this year the prediction, the actual aphid flights were considerably earlier than what the predictions were.
Yes.
Any idea as to why that was, apart from the obvious of a mild winter?
Well, I think the big challenge is actually predictability, because with climate change, it's not a simple matter where we're just seeing a standard routine increase in temperature and across latitudes, etc. We're seeing more extreme events, and those extremes are throwing a lot of the models into somewhat disarray. So it's challenging, and it only takes, on the virus side of things, an initial population to start to spread, and yeah, it's tricky. So I don't know specifically why in this last year we've seen, as you said, the potato leaf roll virus suddenly start to emerge, and aphid flights earlier. But it's an area that we need to do a lot more modelling on, and it's going to handle on. But it's something that we are working alongside with the RIS and the Met Office and other things like the GINS, from multifaceted angles to try and model as much as possible. So yeah.
Yeah, that's because the other one was looking on the insect survey, the Scottish traps. There doesn't seem to be any data after August.
No, so a reason for that is because the RIS is comprised of the 12 English traps. The 12 in England. Meanwhile, the 4 that are based in Scotland are operated by our kind of sister, almost SASA. They actually take their samples to their SASA headquarters and sort them. So we are connected in the sense of the data sharing, but due to a combination of manpower and the way that SASA is structured, they monitor during a specific season. So yes, yeah, that'll be the reason why after August. And I know, for example, the air trap, like AYR, that's has been down for quite some time.
I think the Inverness one is as well.
Exactly. So yes, yes.
Yeah. No, well, it'd be good to have a catch up with you after the meeting at some point, if you don't mind.
That'd be fantastic. I mean, because I'm kind of jumping on you here for a second, but one thing that I'm really keen to pursue is more of a holistic angle of what is actually out there, when and where, because we look at misers and we look at it from one angle of insecticide resistance, but it's spreading such a plethora of viruses, it's got such a range of genotypes that are going on. We need to actually connect the dots so that we can reduce impacts.
There's a huge need within the seed potato industry at the moment for some serious amount of knowledge gap to be filled, which we're looking at the moment. So a conversation will be good after the meeting. So.
Okay, thank you. So there are a couple more questions in the chat, and that probably ought to be the last of the questions after that.
Sure. Hi there, John. I'll do Joe first, just chronologically. So are there any plans to augment the suction traps network with additional traps? So yes, nice, sweet, short answer. We're already in discussion with various other organisations over on the mainland, as well as Chaggers in Southern Ireland, AFBI in Northern Ireland, trying to really bring together back into the fray all of the various such traps that are set up for data sharing and or sample sharing to get that holistic overview. Specifically RIS-wise, we have a lot of quickly deployable traps. So there's some nice concertinaed ones that are almost like a big tube that you can put out where and when, as well as some smaller ones that could be put on scaffolding to reach that 12-meter height that's optimal. But the idea being to actually supplement what we've got to improve the modelling, and especially in areas like Norfolk, East Anglia, where folks like the sugar beet industry are getting absolutely hammered by viruses at the moment, and have been for some time, but our current network isn't necessarily accurate for what is out there. So yes, Joe, absolutely. And then John, hello John, good to see you. So could the predictions have been bad this year because the weather model was parameterized for a 20th century climate? Absolutely yes. So all of the modelling was done 10 plus years ago, and whilst the trends are holding up, it needs redoing, and that is an area that I'd very much like to work potentially yourself. I know we've had some discussions previous, laying the groundwork for better modelling, so that we can actually work on a much more dynamic level on a year-by-year basis. So yeah, we just need more hands on deck to take this amazing resource and data forward. So yeah.
Brilliant. Right. Okay. So yeah, we've just gone past 5 o'clock. So thank you very much again, Lawrence, for an amazing tour de force. Thank you to everybody for joining us this afternoon, and with luck we have recorded it, so we'll load up recording and send everybody the link.
Absolutely. I'll be happy to share the slides as well, especially the ones that I skipped over. So yeah, if anyone wants to review those, just let me know. Yeah,
I can put, if you are, then I'll put them on the same web page as well, Lawrence.
Brilliant.
Yeah.
Excellent. Well, thank you all very much. Really appreciate it.
Thank you very much.